Good morning, my student. We'll talk about chapter 5 sensitivity in the living organism. Lesson 1 sensitivity and living organism. Sensitivity is one of the functions which is done by a living organism. It means how the living organism can respond to or responds to the surrounding environment. It's very important to maintain its life. Sensitivity in animals more obvious than that in the plant. Number one, we'll talk about sensitivity in plants. Number one, response to touch and darkness. There are some plants like the mimosa plant. If you touch a mimosa leaflet, its petiole soon drops as if it has wilted. And also other neighboring petioles soon follow till the effect of is seen in all leaflets. In the daylight, the leaflets are held in horizontal position, and at night the leaflets hang downward and for their upper surface, undergoing three movements. Explanation of this movement. This movement can be explained according to the cell turgidity, as mimosa leaves are compound and pinnate, and each has primary which carry at its end four secondary rachis. Each secondary rachis carry two rows of leaflet at the base of each primary and secondary rachis. And also at the base of each leaflet there is swelling structure called bulbins. When the leaflet is touched or at the night, the primary rachis hang downward. The secondary ones become depressed, and so the upper surface of the opposing leaflet become folded together. The bulbini act as joint in this movement where the lower surface shrink when being touched. As the cell wall of the lower half each bulbins are more sensitive, and this leads to water diffusion to the neighboring tissue and hence the leaflet drop. When stimulus is removed, the cells regain their turgidity and the leaflet open once more. An explanation of phototropism. Boyce and Jensen found that there are scientists found that a vena coleoptile lose its ability to bend toward the light, and its tip, if its tip is cut off from 1 to 2 millimeter of the tip. This ability is restored if the decapitated tip is returned or fixed again to the tip with gelatin. If the tip is separated from the remaining coleoptiles with mica sheet, there will be no curvature, and that indicates that the tip of coleoptile synthesis chemical substance diffuses through gelatin and affects the growth. It can't pass through mica sheet, and these chemicals are termed as auxins. Auxins, the main one is known to be indole acetic acid, and the curvature toward the light is the result of unequal growth of the two sides, which is used due to the unequal concentration of auxins on the two sides of the coleoptile. Also, there is 20 experiments to prove that validity of Boyce-Jensen explanation. Uh, we have also number two, hydrotropism. Hydrotropism. Experiment we have if we germinate a number of seeds in two glass troughs containing equal quantities of dry soil and spray water regularly in the first straw but only as the site in the second trough. Leave these the two troughs for several days. Observation: roots in the first trough grow straight down, while roots in the second trough grow downward toward the water at the sides of the trough. Conclusion: in first trough A, roots grow vertically due to equal distribution of water in the soil around the roots. In the second trough B, the curvature of roots is due to the presence of water at the sides and its absence in the middle. Axons accumulate in the side of the root facing water and inhibit the elongation of cells of that side, while the cells of the far side continue normal growth, which lead to curvature of the root toward water, and the root is known as positive hydrotrophic. Positive hydrotrophic. Okay. Uh, so we can say that the plant stem positive phototropic, and plant root negative phototropic, and plant stem is negative geotropic, and the plant root is positive geotropic. Also, plant root is positive hydrotropic. These all of these due to an equal distribution of oxygens in the upper and lower part of the leaves. Okay. Thank you, my students. Okay, this was the sensation imbalance. Okay.